Yes, dear student, come back to our channel Puttappa Bio Garden. This is the second chapter, second FPC Biology, Sexual Reproduction in Flowering Plants. Regarding the first chapter, we learned about various possible type of reproduction such as asexual reproduction, sexual reproduction in all living organisms in a general. And here this is a specific concept regarding the reproduction, that is sexual reproduction, that confined only to the flowering plants, that is angiosperms. So second chapter it confined to only the angiosperms, and we are going to discuss only the sexual reproduction in angiosperms. So that is the concept here. And as I told you, this chapter carries 7 marks around, and probably 1 5 marks question may also ask in this chapter. So however, this is easy and important. So what is meant by flower? So first and foremost, what is meant by flower? Yes. So as we have studied about the structure of flower in first PUC, we are all familiar with this concept, because flower is the organ of the angiosperm. It is arised from a shoot system usually, and that is why it is a modified shoot. A shoot consists the leaves, axillary buds, branches, flowers, fruits, etc. That is why flower is a modified shoot. It is a modified shoot. Okay, and it is the site for sexual reproduction, as it produces the male and female sex organs. That is why we can define the flower as a modified shoot consisting male and female sex organs that act as the site for sexual reproduction. So that is enough. So therefore, how can you define the term flower? What is flower? What do you mean by flower? Okay, so flower, a modified shoot consisting male and female sex organs that act as a site for sexual reproduction is said to be flower. Okay, so flower, it takes a good position in all human tradition or activities, whatever it may be. Regarding this chapter, so we are going to discuss about the sexual reproduction. That is why I directly go to the concept. As you have studied about the flower in first PUC or in lower classes, a typical flower, look at this. So this typical flower, it has 4 parts, and each part of the flower, it consists, or it is made up of many units. All those units together form that part. Hence each part is called whorl. That is called whorl. Okay, so whorl. Hence a typical flower has 4 whorls. The outermost whorl, look at this. This is outermost whorl, and this is called the calyx. And the units of this calyx are called sepals. Next to this, look at that. That is a very conspicuous and beautiful part of the flower. So that is called corolla. And units of corolla are called petals. ಹೂವಿನ ದಳಗಳಿದೆಯಲ್ಲ ಅದೆಲ್ಲ petals. ಆ ಎಲ್ಲ ದಳಗಳು ಸೇರಿ ಆಗಿರ್ತಕ್ಕಂಥದ್ದೇ corolla, the second whorl of the flower. And come to the third whorl. Look at there, stamens are there. So many stamens are there. These are all stamens together form the third whorl, that is called androecium. Okay, that is called androecium. Here androecium. Okay, androecium. Yes. And the central part, that is having a stigma, style and ovary, that central part is called gynoecium. Okay, so gynoecium. Yes. Hence a typical flower consists 4 whorls, the outermost a calyx. So here suppose this is a calyx. Okay, calyx, and then the second whorl, corolla. This calyx and corolla, they are referred as unnecessary organs or non-essential organs. Why so? Because these two do not take part in sexual reproduction directly. Hence, they are referred as non-essential part regarding sexual reproduction. However, they help in sexual reproduction indirectly. But as far as concerned this androecium and gynoecium, these two are called essential parts of the flower because they directly take part in sexual reproduction. That is why these two are called essential parts. And our sexual reproduction begins from these two sexual organs of the angiosperms. That is the male sex organ androecium. Androecium represents the male sex organ. Gynoecium represents the female sex organ. That is why let me directly go through this sex organ, their structure first. So in the first chapter we explained about the stages, events of sexual reproduction under three headings: pre-fertilization events, fertilization events, and post-fertilization events. In the pre-fertilization events, gametogenesis and gamete transfer. The next one, fertilization, that is syngamy or fusion of male and female gametes. And last one, post-fertilization, that is the formation of zygote and embryogenesis. In the same manner, in the same way, here also we are discussing. Okay. So first, pre-fertilization events. Regarding pre-fertilization events, first we are going to discuss about the gametogenesis. But to understand the gametogenesis, it is better to understand the structure of such male and female sex organs where those gametes are produced. The first male sex organ, that is the androecium. That is why let me go for androecium. Okay. Yes, the androecium representing the male sex organ of the flower, and units of androecium are called the stamens. Okay. Look at the structure of stamen. A typical stamen or a stamen, it consists of three parts mainly. Those three parts, it has a long stalk. Look at this, the long stalk. That a long stalk is called filament. That is called a filament. And then the head region, the terminal part, it has a head region. That a terminal part is called, that lobed structure, that is called anther. And anther, it has many lobes. It may be having single lobe, that is a monolobed or a bilobed. Okay. If it has two lobes, bilobed, or four lobes, tetralobed, like this. Usually, bilobed anther is most familiar and common type of anther. And in our syllabus, we are going to discuss the structure of bilobed anther. And a bilobed structure, that is called anther. And those lobes as well as the filament, they are connected each other by means of a tissue that is called connective tissue. Okay. So that is called a connective tissue. Okay. So it means, so if this is the anther, look at this. So here this is a anther, right? Yes. If this is the anther, then this is the stamen. Look at this. Yes. Now this is the stamen. Then, so here this is a anther. Okay, anther. And here this is filament. Okay, a filament. And the tissue that joins these that the tissue is called connective. That is called connective. Right. Then come to this head region, that is lobe region, that is called anther. What is this anther? Yes, anther. So this is the region where the male gametes or gametogenesis is occurred. So in case of plants, the production of male gametes it referred as microsporogenesis. Microsporogenesis, and production of female gametes, that is called megasporogenesis. So now we are directly goes to this microsporogenesis, right? Microsporogenesis, it occurs inside this anther. To understand that, it is better to understand the structure of this anther. So that is why I am going to take that TS of anther. Okay, transfer section. Okay, so transfer section of the anther. Okay, right. So here the transverse section of an anther is to understand the organization of various group of tissues. So in the inside the anther. So this anther, a bilobed anther, usually bilobed anther. So bilobed anther, it has a two lobes and one lobe, two lobes are there, and one lobe, it consists of microsporangium inside it. Yes, the TS of a bilobed anther, it has a two lobes, 1 and 2. So here 1 and 2 lobes are there. Hence it is called bilobed anther. And anther, as I told you, it is microsporangium. And each lobe, it consists of two microsporangium inside it. These microsporangium are the structures where microsporogenesis will occur to produce the male gametes. That is why such sac-like structures are also called pollen sacs, because they produce the pollen grains. Pollen grains are nothing but male gametophytes. Okay, and each lobe has such a two microsporangium. Such structures surrounded by a sheathy layer, and such structures are called theca. And each lobe has a two theca. Hence it is called dithecous anther. And both the lobes having 4 microsporangium in each corner. Hence it has 4 corners, hence it is, therefore it is called tetragonal, and it consists of 4 microsporangium in total. Hence it is called tetrasporangiate anther. Right? So next this, let us study about the structure of each microsporangium. Right? Regarding this stamen, there is one familiar question in your examination, that is describe the structure of matured anther, or describe the structure of TS of anther ಅಥವಾ microsporangium ಅಂತ 5 ಮಾರ್ಕ್ಸ್ಗೆ ಒಂದು ಕ್ವಶನ್ ಯಾವಾಗಲೂ ಕೂಡ ಕಾಮನ್ ನಿಮಗೆ ಕೇಳ್ತಾರೆ. ಸೊ ಆ ಕ್ವಶನ್ ಅನ್ನು ಕೇಳಿದ್ರೆ ನೀವೀಗ ಫಸ್ಟ್ ಸ್ಟೇಮ್ ಆಂಥರ್ನಿಂದ ತಗೊಂಡು ಪೂರ್ತಿ ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಂ ಸ್ಟ್ರಕ್ಚರ್ ಏನಿದೆ ಅದನ್ನ ಅಷ್ಟನ್ನು ವಿತ್ ಡಯಾಗ್ರಾಮ್ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬೇಕು. ಆದರೆ ನೀವೇನು ಮಾಡಬೇಕು, a typical bilobed anther. Look at this here. This is the TS of anther. Look at this. It has a two lobes. That is one lobe and this is another lobe. Two lobes are there. Hence this is called bilobed anther. And each lobe, look at that. Each lobe having a microsporangium at the center. 1, 2, 3, 4. Those microsporangiums are present at the corner side. And each lobe has a two such microsporangium. And these microsporangium, they are surrounded by sheathy layer or particular specialized coverings. Such structures are called theca. Each lobe having two thecae, hence it is called dithecous anther. In total, 4 microsporangia are there. Therefore, it is called tetrasporangiate. And they are present each corner having 4 corners. Hence, it is called tetragonal anther. Okay, right. So let us study about the structure of microsporangium. Right? Look at this. So here this is the microsporangium. That is the structure of microsporangium. One part of the TS of anther is enlarged here in order to understand the structure of microsporangium. Microsporangium, as I told you, it is the region where the microsporogenesis takes place to produce the male gametophyte called pollen grains. Pollen grains are the male gametophyte which produce the male gametes. Okay, that process of microsporangium occur in this anther, inside this anther, in the microsporangium. And each look at this. Each microsporangium around a spherical structure, sac-like structure. These sacs, they runs along this anther, along these lobes, okay, longitudinally. Suppose you take the transverse section, you look spherical sac-like structures, they run along this anther, along this lobe. Okay, so here they are spherical. Okay, and it is surrounded by 4 wall layers. Each microsporangium surrounded by 4 wall layers. Out of 4 wall layers, look at this. This is outermost one. Outermost one is called epidermis. That is called epidermis. Epidermis. Yes. This is the outermost wall layer of the microsporangium or an anther, young anther. And this epidermis, it is a single celled layer, outermost layer, and it is followed by the next layer, endothecium. Endothecium. Don't forget, look at the next one. That is endothecium. Endothecium. And next to this, there are 2 to 3 layers. That is called a mesoderm. Okay. Middle layers. Okay. Okay. Yes. Middle layers. Middle layers. Okay. Middle layers are there. And next to this middle layers, there is the innermost wall layer. That is a tapetum. Okay. So tapetum. Hence, a matured microsporangium surrounded by 4 wall layers. Of these 4 wall layers, the first 3 wall layers, they take part in protection of the microsporangium. They protect the microsporangium from external stresses, and these 3 layers also take part in dehiscence of the anther during the releasing of pollen grains. So they help in dehiscence. So of these 3, endothecium play a very important role during dehiscence, because this is a water storing or water absorbing structure. That is it absorbs the water, the cells will turgidate increase and burst out and make a slit or opening to release the pollen grains. However, all the 3. And middle layers, after maturity, they become disintegrate or they become destroyed. Okay, disintegrate or disappear after maturity. Next, tapetum. Tapetum, it is the innermost wall layer of the microsporangium or anther, young anther, which play important role in the microsporogenesis, because during microsporogenesis, microspores are produced through meiosis. For that purpose, they require some amount of energy. That energy is supplied by this one. That process of supplying energy for any particular process is called nourishing. So tapetum, it plays very important role in a nourishing. Okay, so nourishing the developing pollen grains, because it has a reserve food material. Hence, it is shown with a dense cytoplasm, and it has multinucleated cells are usually multinucleated cells. Here the nuclei, free nuclear division will occur here. That is why it is multinucleated, dense cytoplasm present in that tapetum, and it has reserve food material. Hence, it acts as a nourishing layer. It nourishes the developing pollen grains. Usually, one familiar question: what is tapetum? What is function? So, our anther with the malfunctioning tapetum results in the loss of microspores, loss of pollen grains. Why? Because it plays important role. Tapetum plays important role as a nourishing layer which nourishes the developing pollen grains. If it is a malfunctioning, if it is any disadvantage or problem, then it could not help in microsporogenesis like that. Okay, so that's why tapetum is very important layer. Don't forget this. ಇದೇ ತರ ಇಲ್ಲಿ ಟಪೆಟಮಿಂಗ್ ನರಿಶಿಂಗ್ ಇದೆಯೋ ಇದೇ ಈ ಚಾಪ್ಟರ್ ಮುಗಿದ್ರೊಳಗಡೆ ಮತ್ತೆ 3-4 ಇನ್ನು ಹ್ಯೂಮನ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಅಲ್ಲೂ ಕೂಡ ಇದೇ ಕೆಲಸ ನರಿಶಿಂಗ್ ಮಾಡ್ತಕ್ಕಂಥ ಬರ್ತಿರ್ತವೆ, ಎಲ್ಲವೂ ಕೂಡ ರಿಲೇಟೆಡ್ ಇರ್ತವೆ. ಸರಿ, ನೆನಪಿಟ್ಕೊಳ್ಳಿ. ನೆಕ್ಸ್ಟ್ ಕಮ್ ಟು ದಿಸ್ ಇನ್ನರ್ ಟಿಶ್ಯೂ ಮಾಸ್ ಆಫ್ ದಿಸ್ microsporangium, right? Yes, inside this microsporangium there is a homogeneous tissue consists the sporogenous tissue material. It's a sporogenous tissue having all the diploid cells which are homogeneous in condition, and this sporogenous tissue in young anther, it undergoes development, and after the maturity or complete the growth, each sporogenous cell, it acts as a spore mother cell. That is, here they are called pollen mother cells. Pollen mother cells are microspore mother cells. Okay, pollen mother cells. These pollen mother cells, after maturity, they enter into microsporogenesis. This is the structure of a microsporangium, and this is the structure of young anther. So for this purpose, you have to draw the diagram. So anther, the TS of anther, and structure of microsporangium. These are all essential. After this, what is microsporogenesis? So after this, we are going to study about that is a gametogenesis, pre-fertilization events or gametogenesis. So here, production of male gamete is called microsporogenesis. Don't forget, in case of a human reproduction, the third chapter, it is called spermatogenesis. Here, microsporogenesis. So what is this microsporogenesis? This is the process of formation of a haploid microspora from diploid polar mother cells. From diploid polar mother cells through meiosis in the microsporangium of anther is called microsporogenesis. So after maturity of these polar mother cells, they undergo meiosis, and each polar mother cell, it produces 4 haploid cells. Those 4 haploid cells are called microspores. Initially, they attached in a group. They arranged in a group, and that group consists of 4 microspores together called a microspore tetrad. Look at this. So microspore. Suppose this is a polar mother cell. Look at this. This polar mother cell finally it divides into 2 cells, and then again into 4 daughter cells. These 4 daughter cells they remain together. Look at this. So they remain together like this. Yes. So yes. Okay. So this is one microspore tetrad, right? So one microspore tetrad. This is one microspore, 2, second, a third, and fourth. At the initial stage, they attach, they cling to each other. Okay, they attach together and remains in a group that is called a spore tetrad. So this spore tetrad, it remains up to the complete maturity of these microspores. As they mature, the anther it dehydrates. Okay, as it dehydrates, these microspores they become spherical in shape, and they dissociate each other. They release each other as a one pollen grain. So one microspore. Such a thousands together, several thousand microspores or pollen grains are produced in these microsporangium. Okay. So after dehiscence, these pollen grains they released from the anther. Okay. So this is a microsporogenesis. Yes, friends. So it is better to understand this through the textbook also. ಸೊ ಎಷ್ಟು ಸಾಧ್ಯ ಆಗುತ್ತೆ ಅಷ್ಟು ಈ ವಿಡಿಯೋನ ಮತ್ತೆ ಮತ್ತೆ ನೋಡಿ, ಬೇರೆ ವಿಡಿಯೋಗಳು ಸಿಕ್ಕರು ನೋಡಿ, ಏನೇನು ಕ್ವಶನ್ಸ್ ಇದೆ ದಯವಿಟ್ಟು ಕೇಳಿ, ಹಾಗೆ ಟೆಕ್ಸ್ಟ್ ಬುಕ್ಕನ್ನು ಚೆನ್ನಾಗಿ ಫಾಲೋ ಮಾಡಿ. ಇದಾದಮೇಲೆ ನಿಮಗೆ ಒಂದು ಹೋಮ್ವರ್ಕ್ ಕ್ವಶನ್ ನಾನು ಕೊಡ್ತಾ ಇದ್ದೀನಿ. ಅದು ಡ್ರಾ ದ ನೀಟ್ ಡಯಾಗ್ರಾಮ್ ಆಫ್ TS ಆಫ್ ಯಂಗ್ ಆಂಥರ್. ಯಂಗ್ ಆಂಥರ್ ಅಂತಂದರೆ TS ಆಫ್ ಕಂಪ್ಲೀಟ್ ಆಂಥರ್ ಹೋಲ್ ಮೌಂಟ್ ಅಂತ ಫಸ್ಟ್ ಹಾಕಿ ಚಿತ್ರ ಅದು. ಆಮೇಲೆ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಮೈಕ್ರೋಸ್ಪೊರೆನ್ಷಿಯಲ್. 2 ಡಯಾಗ್ರಾಮ್ ಚೆನ್ನಾಗಿ ಪ್ರಾಕ್ಟೀಸ್ ಮಾಡಿರಬೇಕು ನಾನು ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ಅಷ್ಟೊತ್ತಿಗೆ. ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು.